Hello everyone, I am Dr. Sarita. I welcome you for this session on microbial degradation of cellulose. Lignocellulose is a combination of cellulose, hemicellulose, and lignin. Lignocellulose is a predominant biopolymer of plant cell wall. Cellulose is an important structural component of primary cell wall of green plants, algae, and oomycetes. Application of cellulolytic microorganisms and their enzymes for the pretreatment of lignocellulosic material is found to be the best method in various industries for the production of primary and secondary metabolites. Cellulose is an organic compound belonging to the category of polysaccharides. It is abundantly present in plant and bacterial cell walls. Cellulose plays an important role in the structure and strength of plants. It also finds great importance in the industry. The purest form of cellulose is cotton that contains around 98% cellulose. Cellulose is a linear polymer made of glucose subunits linked by beta 1,4 glycosidic bonds. Each glucose residue is rotated by 180 degree to its neighbors so that the basic repeating unit is in fact cellobiose. Cellulose chains are oriented in parallel and form highly ordered crystalline domains interspaced by more disordered amorphous regions. The role of cellulose is exclusively structural. The mechanical strength of cellulose is particularly obvious in the case of wood and textile fibers, which consist of the walls of elongated empty cells. Cellulose chains form numerous intra and intermolecular hydrogen bonds, which account for the formation of rigid, insoluble microfibrils. The high tensile strength of cellulose enables plant cells to withstand osmotic pressure and is responsible for the resistance of plants to mechanical stress. Endoglucanase usually attacks amorphous areas of cellulose. The random attack of this enzyme on internal bonds of loosely bound amorphous areas of cellulose creates new chain ends. These new chain ends are then easily attacked by other types of enzymes. The highest activity of this enzyme usually occurs against soluble cellulose forms or acid-treated amorphous cellulose. The function of exoglucanases to produce glucose or cellobiose units by attacking the reducing or non-reducing end of cellulose chains. Endoglucanase is different from exoglucanase because it is usually very active against crystalline cellulose substrates. Beta-glucosidase can hydrolyze cellobiose to glucose from the non-reducing ends, and it is inactive against amorphous or crystalline cellulose. Although an exact mechanism is not yet finalized, fragmentation of cellulose aggregations into short fibers has been observed and reported during the beginning of cellulose hydrolysis prior to releasing any detectable amount of reducing sugars. This is known as amorphogenesis. Complete degradation of glucose, which is ultimately converted into carbon dioxide and water under aerobic conditions and into carbon dioxide, methane, and water under anaerobic conditions. Topsoil is the most important aerobic biota where cellulosic waste accumulate. Among these, wood is the component most slowly attacked by cellulolytic microorganisms due to its high content of lignin. The fungus Trichoderma resi, an aerobic and highly cellulolytic deuteromycete, is probably the microorganism whose cellulase system has been most thoroughly investigated. Among aerobic cellulolytic soil bacteria, several species belonging to the genera Cellulomonas, Pseudomonas, Thermomonospora, and Microbispora have been studied in detail. The process of cellulose degradation is called cellulolysis. Cellulase is not a single enzyme, it is a group of enzymes which is mainly composed of endoglucanase and exoglucanases, including cellobiohydrolases and beta-glucosidase. Fungi, bacteria, and actinomycetes are recorded to be efficient cellulase enzyme producers in the natural environment. These microorganisms must secrete cellulases that are either free or cell surface bound. The classic scheme for cellulose degradation involves the synergistic action of three classes of enzymes. Endo 1,4 beta glucanases randomly cleave internal bonds in the cellulose chain. These enzymes may be non-processive or processive. In processive enzymes, enzyme substrate association is followed by several consecutive cuts in a single polysaccharide chain that is threaded through the active site. Exo 1,4 beta glucanases attack the reducing or non-reducing end of the cellulose polymer. Processive exo 1,4 beta glucanases are referred to as cellobiohydrolases. They are among the most abundant components in natural and commercial cellulase mixtures. Beta-glucosidases convert cellobiose into glucose. Cellobiose is the major product of endo and exoglucanases. Cellulolytic microorganisms were isolated by serial dilution technique. Then the pure isolates were transferred on carboxymethyl cellulose agar plates and incubated at 30 degrees Celsius for 2 to 3 days. After incubation, the plates were flooded with 1% Congo red and the plates were kept undisturbed for 20 minutes at room temperature. Then the plates were thoroughly washed with 1 molar sodium chloride solution. A clear zone around the colonies were evident for cellulase producers. Cellulases are currently the third largest industrial enzyme worldwide by dollar volume because of their use in healthcare, textile, pulp and paper, detergent, food and beverages. Its wide application in coffee processing, winemaking and fruit juice production is related to food and beverage segment. In other industrial applications, it is broadly used to produce laundry detergents and cleaning and washing agents. Cellulase is also being highly recognized as an effective alternative to available antibiotics for the treatment of biofilms.